Our next presenter is Dr. Jared Miller. Dr. Miller grew up on a farm in Maryland before going to Virginia Tech to pursue a bachelor's in environmental science and a master's in crop and soil environmental sciences. He finished his graduate work at the University of Kentucky with a PhD in soil science. Before coming to work for the University of Maryland Extension on the Lower Eastern Shore, he held postdocs in the Forestry Department at the University of Kentucky and the Agricultural Research Service in Florence, South Carolina. Today, he will be presenting his presentation titled Denitrification in Natural Converted and Restored Wetlands of the Mid-Atlantic. Welcome, Dr. Miller.
All right, thank you. So I'm covering the denitrification portion of this talk as well as some of the research that I've done in the past dealing with it. Denitrification is a reduction reaction. It's a redox reaction. If you remember from chemistry, reduction reactions like iron 2 and iron 3. If you add electrons to iron 3, you reduce it to iron 2. So nitrate in the soil is an oxidized form of nitrogen. And denitrification is primarily a biological reaction where in their respiration, as they're consuming organic matter, they have these excess electrons as they're producing energy, and they like to deposit these on typically oxygen in an oxidized environment, but when they don't have oxygen available, they'll use nitrogen and other inorganic compounds. So nitrate in the soil can be reduced all the way to dinitrogen gas. So this is a pathway of loss for nitrogen in these soils. So when denitrification occurs, and you can see several steps here, all the way to, in the blue here, you have the gas stages. So once you get to these stages, these can actually leave the soil, and it's a different loss mechanism. Denitrification primarily occurs in low oxygen, which would be saturated soils, because microbes are going to prefer oxygen as their electron acceptor because they get a lot more energy from it. The main issue we have... Yeah, can you hear me?
Excuse me, Dr. Miller? I'm sorry.
Yeah, sure, no problem.
Yeah. Can you speak a little bit closer to the mic, please?
Yeah. All right.
Sorry.
One of the main issues environmentally, because dinitrogen gas right here, the atmosphere is about 80% nitrogen. This isn't a problem. But as Trish mentioned before, N2O can be a greenhouse gas and also affect ozone. So if this reaction is incomplete, you produce N2O. So this is a simple overview of the nitrogen cycle, and here would be denitrification over here, typically biological in nature, and taking nitrate and reducing it into atmospheric nitrogen. So who causes denitrification? It's anaerobic bacteria. And anaerobic means no oxygen or low oxygen. So some of these microbes that do denitrification, they can operate in both aerobic and anaerobic environments. But for denitrification to occur, it's usually an environment that has less than 10% oxygen. So in soils, this would be an environment that's saturated. Oxygen diffuses into soil about 10,000 times slower through water than through air. So once your soil starts to have moisture in it or becomes saturated, you end up with less oxygen. These microbes themselves are mostly heterotrophs, which means they obtain energy by oxidizing carbon, which means they're like you, they're eating organic compounds to produce their energy. They're not using photosynthesis. So the organic matter and carbon source is important in denitrification. The overall reaction is actually, it's reducing nitrogen, but it's also releasing the oxygen from the nitrogen compounds to be reduced. There is an order of preferred reduction in the environment. So over here, grab this arrow, the oxic, where you have oxygen. Again, oxygen gives microbes the most bang for their buck. If oxygen is available, you have a larger energy reaction and you have quicker energy reactions. But once they use up oxygen, once you get to these environments where you have less oxygen, saturated ones, they start to find other electron acceptors that are out there in the environment. from nitrate all the way down to carbon dioxide. So when you get into an environment like a bog down over here, you might start to sell sulfur and methane, which is usually it means you're in a very reduced environment. So nitrate's at the top of the scale, which means it still has quite a bit of energy compared to some of these other elements. The issue with this scale is a lot of people think that it's just going to happen in this order, but because soils can be complex, it's not necessarily going to occur in the order given, or you can have both reactions occurring in the same environment. So over here this is actually a slice of soil. So you can see the pore space, along in here, and you can see the soil solids. In your soil, even though nitrification here occurs in oxidized environments and denitrification occurs in reduced environments, they can occur at the same time in the soil because of how slow oxygen might diffuse through. So over here where you have large pore space, and so you have oxygen coming in from the surface of the soil, you can have nitrification occur. But in these smaller pores, if you have saturation, and your soil doesn't have to be underwater, they can just be saturated right in this section here in the small pores, you can have denitrification occur side by side. So measuring the amount of denitrification and nitrification going on in the soil can be difficult because they can occur at the same time. Now, if you have about 6 inches of water sitting on top of the soil, you can assume that all these pores are saturated and mostly denitrification is occurring. So it's the same thing with that scale. Although nitrate would be preferred, if nitrate is only available in this section of the soil and iron's available over here, then the microbes are going to use whatever element is available as their electron acceptor, as they're respiring, as they're eating. Even though this nitrate might be available to a microbe, that might be like driving across the country. It's not going to know it's there. So you can have reduction of both of these elements occurring at the same time. So organic matter is a microbial energy source. It's what they're eating to produce their energy. It's what Trish mentioned, those carbon bonds, the electrons in there. They help them get the energy. As an energy source, you can split up organic matter into 3 parts. There's a lot of different ways to do it, but one way to simply describe it is fulvic acids, humic acids, and then human. Fulvic and humic acids are usually found in fresh organic matter, and they're going to have more of the N, P, and K inside of them. So in fresh organic matter, you have a more accessible energy source that has all the nutrients they need, because they don't just want the carbon. Like Trish was mentioning about immobilization and mineralization, they want a range of nutrients. They're just like us. So if you're not adding fresh organic matter, over time you can actually lose this as microbes break it down and end up with more human, which is older, it has more carbon, and has less nutrients. So this can kind of change your environment where bacteria are going to prefer to have fulvic and humic acids. You might have more fungi when you're dealing with human. So it's not just having organic matter as an energy source, but also the quality and the type of organic matter you have. And this comes back to the C to N ratio that Trish mentioned before. And for mineralization and immobilization, like the rates she talked about, 18 to 25 to 1, if you have something like wood chips that say it has a C to N ratio of 80 to 1, that means it has 80 times more carbon than nitrogen. So this bacteria is going after the carbon source as its energy. It also needs some nitrogen, but there's not enough nitrogen in here, so it steals it from your soil. So that's the mineralization, immobilization. For denitrification, C to N ratio is more important in terms of what's in the soil. It's not the material you're adding, but the overall C to N ratio of the soil itself. So for denitrification, both nitrate and carbon are important. If your soil C to N is greater than 25, it means it has more carbon, more dinitrogen gas is produced. That means the reaction goes all the way to completion. When your soil C to N is less than 25, you have more incomplete. And all this means, so here's your complete going all the way to N2, is if you have a material that has enough carbon, it has enough electrons, it has enough energy, then you reduce it all the way to dinitrogen gas. But if you don't have, when you have a lower C to N, that means you have more nitrate than you have carbon as the energy source, you end up with incomplete. So while some might go all the way to N2, some of this will come off as N2O. So denitrification can be incomplete and produce N2O, or be complete and produce N2. Besides carbon and nitrogen and moisture, temperature is also important. This should be right here, that should be 25 to 35 degrees Celsius, which is about 70 to 95 degrees Fahrenheit. So for temperature, microbes are living things, and moisture affects them, but so does temperature. And typically for the research that I found when you're out here on the Delmarva, when you get past about mid-October, they pretty much shut down. They're still in there, they're still active, but at much lower rates and lower numbers. So temperature is a factor in denitrification. It can occur from close to freezing temperatures all the way to very warm temperatures, but it'll be lower rates. You'll see your maximum going from about 25 to 35 degrees Celsius. pH also has an effect. It's not the strongest effect, but if you have a low pH, less than 5, it favors N2O production and inhibits rapid denitrification. So overall, denitrification, in summary, is affected mainly by oxygen depletion because this is an anaerobic reaction, but that oxygen can vary across your soil porosity. How quickly is it diffusing through the soil? How quickly is it lost? What's your pore space, macropores, micropores, soil texture, your bulk density? That's all going to have an effect on your oxygen in the soil. And then after that, your nitrate concentration, because this is denitrification, so how much nitrate in your soil is going to affect the reaction. So that can be related to your C to N. And then the quality and quantity of carbon. It's not just the amount of carbon, but what's the quality of it? How much fulvic and humic acid? Is it a food? Is it a balanced food source for these microbes? And you can generally relate that to C to N. And then temperature and pH both have their own effects, but these top three here are the most important when considering denitrification. So the obvious issue here, when you see all 5 of these things, is how are you going to predict denitrification in a soil when you have so many factors affecting it? Now, saturation across the pore space and nitrate and carbon can all vary within a couple inches and a couple feet in soil. Temperature might be a little more consistent and pH might be a little more consistent, but the variability in these in soils will make a large variability in denitrification across a landscape. So how do you measure denitrification? One way they came up with in the 70s was to measure using this thing called DEA, or denitrification enzyme assay. So these microbes in the soil, they're using enzymes to cause this reaction to go to completion. And you can measure the activity of these enzymes through this process. The issue with DEA is if you go all the way to completion, you're getting dinitrogen gas. Well, if your atmosphere is 80% nitrogen, and you only have 1 part per million change in nitrogen coming out of your soil due to soil microbes, it's hard to measure that against a background that's so large. It's like a needle in a haystack. But what we can measure is N2O, because it's at much lower concentrations in the atmosphere. We can use gas chromatography and measure N2O coming out of these soils, but that only gives you the incomplete. So what they figured out is we measure N2O by gas chromatography, and then if you add acetylene to these soils, it blocks this final conversion. So you have to take 2 measurements. First you measure the N2O that's coming off of the soil, and then you come back and you add acetylene and you prevent all of this from forming, so everything comes off as incomplete. And that will be your total, or your complete denitrification. If you simply subtract incomplete from total, you get how much is coming off as N2. So that is the primary method through which we measure in a lab setting denitrification. You can choose to add nitrogen or carbon as a source to maximize potential, because this is just a potential in a soil. It doesn't mean it's actually happening when we measure this. Because nitrate's important, you'll want to know, well, if more nitrate comes into the soil through runoff or through fertilizer additions, will that increase DEA? And the same thing, if we add more carbon, will that increase DEA? So you can add those in your experiments. You can also measure in the field. One way they do it is they use these static chambers here. And a typical way to do the static chambers is you just place it in the soil. You can't see from here very well, but that little dot right there, you can actually pull gases out of there, take them back, and measure them on the GC. There's some issues with transporting them back from the field. They also have this machine called a photoacoustic gas analyzer, which we got to use at the ARS. It's a really interesting piece of equipment. It's been around since I think the 70s or maybe before that. It pulls in a gas sample. It uses light, and that's the photo part of the name. So light can excite gases and cause them to expand. And they flash the light on and off. And so it causes these gases to expand and contract. And inside they have microphones that pick up the noise of that gas expanding and contracting, and they can actually get a concentration very accurately of N2O CO2. Now there are some problems in the field with humidity with this machine, I can tell you, but you can get very accurate numbers if you know how to use it. The issue is over here, some N2O can be coming from nitrification and some can be coming from denitrification. So you can't be sure in one sample how much is coming from denitrification. All you can do is relative comparisons across maybe a wetland landscape to determine. So that's in general what denitrification is and how we measure it. And here were two of the projects that I got to work on when I was at the ARS in South Carolina. The first one is the SEEP project. These were mainly, for me, it was in the mid-Atlantic states, including Maryland, Delaware, Virginia, and North Carolina. And then in the Florida Everglades, we did some work with sugarcane and water table management. So the SEEP project is the USDA Conservation Effects Assessment Project. It's about observing environmental effects of conservation practices. So we go out and look at restored wetlands for their how they've restored them and their ecosystem functions. How well are these restorations working? Are they going the direction they want them to? And this is supposed to help the USDA guide future efforts for conservation policy. So if one restored wetland or one method is working better than another, it can guide future efforts. This was a multi-agency effort. Again, they're doing these across the country. Ours was in the Mid-Atlantic. We were working with the NRCS. Of course, I was at the ARS. The USGS was part of it, and even the University of Maryland was doing some of the soil mapping and plant analysis. But again, I worked at the Agricultural Research Service, and down there, our part was to do denitrification and gas emissions. USGS did some work with water. Some ARS in Penn State, they did some nitrogen and phosphorus. This was the region we worked in. So I was all the way down here in Florence, South Carolina, and we looked at soils that span the eastern shore, from Delaware over into Maryland, the middle coastal plain of Virginia, and then the only sites we could get access to in North Carolina that had significant restorations were in the Pecosins, which are flat organic soils. So they didn't quite match the two regions up here. It's a good long drive from Florence. It's about 5 hours to there and 8 hours to there. So collecting DEA over 2 years takes quite a bit of time. We had 48 sites across the 4 states. 21 were in Maryland, 7 in Delaware, and 11 in Virginia. And part of the reason there were so many in Maryland is there were more restorations done or more farmers that allowed us access to their land, because you have to get permission to come out and sample these. The data I'm going to show you does not include the stuff from North Carolina, which we just put out in a separate paper, because the soils down there are highly organic relative to these 3, and they're very different sites. We were looking at 3 site types: converted, restored, and natural. So we were looking at depressional wetlands, not wetlands along rivers and not wetlands affected by the tide, but depressions in a landscape that collect water. So there are three types. The converted is the drained ag land. So this is land being farmed, and converted is just a short name for prior converted, meaning it was drained prior to laws in the 80s so that they are allowed to still farm it. It's grandfathered in. Restored, this would be the NRCS restored wetlands where they've come onto farms or other lands and restored some land that had been drained. And then what we called natural, and sometimes I just call it forested. We can't really say that it's never been disturbed, but we were pretty sure it hadn't been drained. But at some point in the last 100 or 200 years, you could tell that they had at least been logged. At the time, these NRCS conservation practice standards were called 657 wetland restoration and 646 shallow water management. So those were mainly the restored types we were looking at were these two. This is some pictures from actually the Virginia sites. So here's your converted. This is just some cotton fields with drain tile underneath them. Here would be a natural site, what we call forested. So you can see it's a forested wetland. You can tell by these trees that they're not virgin timber. So in terms of natural, it's just a relative comparison. And this down here is actually a restored wetland in Virginia, where they have a berm back here and a water control structure. And they just put it in place to hold water. As you come up out of this wetland and to the edge, you actually come into agricultural lands because these were usually on the edges of fields. So a lot of these, part of their function is to receive drainage from ag lands and try to intercept and process the nutrients. But they are depressional, so you're coming up from the edge of a bowl down into the bottom. Now I'm not talking about the North Carolina sites, but I thought they were interesting, so I leave the pictures in. In North Carolina, it's very flat landscape. These precosins are highly organic soils. The only water in and out is usually from rainfall and evaporation. They do have drainage ditches down in here in their ag land, just like on the peninsula here. And their natural wetland there was actually the Pocosin Wildlife Refuge and was a lot more established. These soils are so different in their in how they work with denitrification, they can't be included with this data. So again, these were depressional wetlands, and we were doing denitrification, and since it's so hooked up to the moisture in the soil, we actually had zones, topographical zones that went from the upper edge of the bowl down into the bottom. So we used GIS to create 4 different zones. It was easy with GIS for it to pick the lowest point, but we had to, through either field observation or aerial photos or topographic maps, we had to pick the edge of this bowl, and then we let it separate out 4 different zones. Other people that sampled on this project would have had their own methods, but since denitrification is so dependent on moisture, we wanted to look from the upper edge down to the bottom wet spots. What's the difference in denitrification? Because it's also going to vary by temperature, we sampled each wetland 3 times over a 2-year period. We got out there 3 times, at least spring, summer, fall, as best we could to try to get temperature variation too. Because you can't just have one number for DEA because so many factors affect it in its variation. So here's the picture of that Virginia wetland again. And these aren't where the points were, but you can kind of get a good idea that 4, whenever you see the number 4 in here, that's your driest point. It comes down into 3, 2, and 1. So you're going from a drier to a wetter point. And overall, this worked pretty well. And so for a side view, so you understand what I mean, this is depressional wetland. It does have a drainage point, not like the Pecosins. But 4, 3, 2, 1, you come from the upper edge down into the bottom. So you're going from drier to a wetter point, which should increase denitrification. In our lab, for our DEA analysis, we had your typical, your incomplete, where you're measuring N2O, and then we did a nitrate addition. So if nitrate comes into the system, how much of it would be denitrified? How much would be processed? Would it increase reaction? Then you add that acetylene, and that's the one that blocks it going from N2, so you get that total, which is measured as N2O by gas chromatography. You can add glucose, but we didn't in these experiments. My former boss had found that in riparian buffers that glucose didn't affect soils there. We also used the photoacoustic gas analyzer. It can actually measure 6 gases at once, so we actually could get N2O and CO2, so there's your microbial respiration. And finally, we did DNA extraction, where we looked at the NAZ gene. There are actually 4 enzymes that occur in the reaction going from nitrate all the way to N2 in the reduction. But this one gene here, NAZ, it encodes, within these microbes, it encodes the enzyme that converts N2O to N2, so that final step. So if you measure a lot of this gene, you know that those microbes are active in the soil. So it's another way to get at these final results. So I'd like to go over all the soil properties, which will help explain some of the differences in the results we saw, and also give you a good idea of what's going on in some of these different types of wetlands. So first off, the carbon percentage in these soils. So you can see over here I have your 3 management types, your natural, your NRCS restored, and this is your ag land, this prior converted. Then you go your elevation, 1 is your wettest, 4 is your driest. It makes sense that your carbon is highest in the natural and the wettest point. So you have a natural forested soil, it's depositing leaves, they're hitting a very wet spot and they're not being broken down, you have a lot of residual carbon left over. You come to converted, 4, this is your driest, this is the upper edge of that depressional wetland, and that's actually your lowest carbon. So you see these letters along the side if you haven't seen statistics before. If you see something with the same letter, like you see 2.0 and 2.1, they're the same. Statistically, found no difference in them. But A and G are very different. So carbon is highest in a forested soil where it's wet. Carbon is lowest when you come up to the edge of a converted dry ag field. Nitrogen follows the same thing. Your total nitrogen, in general, it decreases as you come up out of that depression in a natural. It's highest, though, in the natural versus all of the others. And it's higher, the lowest number is, once again, at the upper edge of that converted depressional wetland. C to N ratio overall follows the same trend. You have your lower C to N's, your higher C to N's are actually in the natural, and your lowest are in the converted. And your C to N actually rises as you come up out of here, so you're losing more nitrogen than you are carbon. pH follows what you'd expect. You're in a forested system. You have lower pHs due to those organic acids. You have your higher pHs down here in the converted where it's going to receive lime. Same thing with moisture. Your wettest point is the lowest spot in the natural. Your driest point is the upper edge of that converted. Now, data like this makes sense, but it's really nice to see as a scientist because it means that you at least set up your experiment correctly because your moisture follows what you wanted it to. Soil temperature, when you're under forest cover, there's not much difference. See all those D's? There's not much difference. You have your coolest temperatures and your warmest temperatures is up on the edge of that converted field. Calcium. So calcium would be a relic of liming. You can see that in this natural soil, there's not a whole lot of calcium left when compared to converted. So here in this farmland, they're going to be liming, adding calcium and some magnesium to keep their soil pH up. Restored sits right in the middle. So this kind of gives you an idea that it hasn't reached a natural state, but if it had at one time had the same amount of calcium that the ag fields up here did, that it is losing it over time. Magnesium is fairly similar across converted and restored, and it's lowest in natural. So again, this is probably a relic of adding magnesium as a lime. Potassium, I think, shows itself being leached easily out of the soil. The converted, again, this is ag land. They're going to add potassium to fertilize the crops, but the restored has already lost potassium and is similar to natural. The only thing about saying that is I don't have any data from before we sampled these. So for all I know, these restored sites never had a lot of potassium, but you can't assume from this comparison that it's possible that if they had the potassium, they lost it. I just can't say it definitively without seeing data from these soils prior to them being restored. So then you get into the anions. Nitrate being the most important because we're talking about denitrification. Now, if you remember, I said that the natural soils, the forested soils, had the highest nitrogen, but they actually had the lowest nitrate. So they had the highest nitrogen because of all the organic matter that's still there. But when you come down here, farmers are going to be adding nitrate to these soils to fertilize their crops. So overall, it makes sense that this ag land is going to have higher nitrate. Even though it has lower total nitrogen, it has more nitrate. And since nitrate is a primary component of denitrification, it's not a bad assumption to think that you might see more denitrification here because nitrate's available. And another point, I think, is right here on the edge of the restored, you see that there's more nitrate than in this natural and even some parts of the restored here in the lower positions. And I think this might be because the restored wetlands are usually right along the edge of ag land, so they might be receiving some nitrate, but then you see it drop off quickly when you go into the wetter portions, which might mean that denitrification is occurring and these wetlands are performing like they're supposed to. Sulfur I only leave up here for anybody who is in agriculture because you can see that the natural sites have more sulfate than converted, and so it might indicate for any farmers out there that you want to watch your sulfur levels, because in a natural soil we see more sulfur than we are in converted, so we might be losing that. And then phosphorus, the same thing. There's higher phosphorus in converted ag land than in a natural soil because they are adding it for fertilizer crops. So that was all the soil information that kind of puts together what kind of picture you might expect to see when dealing with denitrification. These graphs are from the paper that we published. It is the denitrification across converted, restored, and natural wetlands. The black here, that's your N2O. The white here is N2, and together they make up your total. So when you are up here, you're looking at straight soils. Down here we've added nitrate plus NO3. So we added nitrate to these soils to push DEA as high as we could get it. The point right here is that there's really no difference in incomplete, AAA. So the incomplete coming off all these different land types is the same. There's no difference in N2O, which is a good thing. But we do see a difference in the total. And if you remember, in the converted, it had the highest nitrate. So you're actually seeing more DEA in this site, even though the converted is probably drier than the natural. There's more DEA going because there's more nitrate available. When you add nitrate to these soils, it flattens out. Everything becomes equal. So by adding nitrate to restored and natural, you actually erase the differences. So what that might indicate is that these soils over here, even though they don't have the denitrification occurring, they have the potential. They have the microbes, they have the capacity. They just need the additional nitrate. So if they are along the edge of a converted field, if there is some nitrate that comes off, they can handle the denitrification. But you also, when you pump that much nitrate in there, you increase the incomplete a lot. Now this is just a lab study. This doesn't mean it would happen in the field. You're kind of forcing this reaction. So this is elevation 2. This is when you're coming up. So the last one we were looking at was the wettest spot. This spot is coming up a little bit. Same results. Converted has the most DEA total. Restored and natural, when you add nitrate, they catch up. So the two wettest spots have the potential to denitrify, but where you have the nitrate, you see the most initial activity. Elevation 3, we're getting a little drier. Here, you see that the restored and converted are similar to each other, and natural is the lowest. But when you add nitrate, they all catch up to each other, and all pretty much the same. When you come to the driest position, elevation 4, is when you really see something interesting. Again, converted and restored are the same. There's nitrate available there. They're wet. Denitrification is occurring the same way, a little bit lower rates. Natural is very low at this point, up at elevation 4. And when you add nitrate, natural doesn't catch up. So that might indicate that it doesn't have the potential at its upper edge to denitrify. This is the emissions data. I'm very cautious when showing any emissions data at all to anybody because People like to use the absolute values and say it means something, where I really think you need to compare the relative values, because this data is so variable. It's so variable that we never really found any differences across all these site types. Your N2O here is in black, and here's your CO2 in gray. CO2, which represents microbial respiration, how much they're eating, is actually pretty consistent, and the values don't vary too much. But nitrous oxide, because so many of those factors can vary across the soil, from temperature to nitrogen to moisture, the variation is so great here that we couldn't find any comparison between them. I can't say anything significant. So while natural has the lowest N2O produced, I can't actually say it's statistically different. And down here, this is by temperature, spring, fall, spring. So we're looking at seasonal changes. Again, nitrous oxide in the spring, the variability was so high right here that even though the variability was low in these two, we couldn't find any differences statistically, which just means we probably need to sample more. But you can see that overall carbon dioxide is higher in the spring than it is in the fall, and that's just something to do with temperature and how active the microbes are. This is the DNA extraction. This data is a little more reliable than field emissions of gases. So this right here is a total count. So when you see a higher number, that means there's more of the enzyme. So for the converted, and here again is your ABC for your statistics, converted soils had a higher count of that NAZ gene, that enzyme that converts N2O to N2. So these converted soils have a lot higher microbial population than restored and natural, which again I think could be related to the actual nitrate concentration that's already present. When you look at moisture gradients, which averages all these together, your wettest spot has the highest count, while your driest spot, what they're calling the highest, has your lowest count. So it follows the moisture gradient pretty well. When you go from wet to dry, you lose your denitrifiers. So higher nitrate, higher moisture, it makes sense. It follows what's already known about denitrification. When you split these out, when you match the management with the elevation, you see what actually still dominates it is, even though converted is drier overall, it still had a higher count than the wetter restored and natural sites. So even though we consider moisture to be the most determinant factor in denitrification, in these systems that we were looking at, it was actually the nitrate concentration that seemed to be driving enzyme production. So in general conclusion, for this project, with the wetlands that we looked at, is that I do think that restored wetlands are similar to both natural and converted, so they're kind of sitting in between. They retain some calcium magnesium from liming. They haven't quite built up the carbon and nitrogen yet, but I do think they will continue to move towards a natural state as they are. For DEA, restored wetlands are like natural when you reach the bottom. They act more like converted at their upper edges, and this is where I think you're seeing some nitrogen enter from the edge of these fields, but it gets denitrified before it reaches the middle, so you don't see as much activity. So I do think, with what we're looking at right now, that they are performing their function in intercepting nitrate. Now, I can't prove that. It's just what I think from the data that we've seen. So that was the Mid-Atlantic, which were depressional wetlands. They were typical soils that had maybe 2 to 6% organic matter, depending on where you were looking. We also had a project that we looked at in the Everglades, which were highly organic soils. So these soils down there, being organic, when they were exposed to oxygen, so they built up over time, because when they were saturated, you had slower breakdown, like we discussed before, and the organic matter builds up. When you expose it to oxygen, you can speed up the reaction, and it's actually pretty much, in a sense, evaporating off of there as carbon dioxide. So this is a post that they drove in the ground. I think it says 1924 here. But this was the surface of the soil in the 20s. This was the surface in 2012. So what you're seeing is, over time, some of it could have been runoff, but you're just seeing a loss of this soil over time, pretty much in one sense through volatilization, evaporation, microbes turning that carbon into CO2. So because of that and because of the loss, and this is they do a lot of sugarcane down there, they're looking into controlling the water table level to reduce losses of nutrients as well as reduce the mineralization of that organic matter. And down the ARS has a research station down there, and they have these lysimeters, which Trish had mentioned the pan lysimeters earlier. And so they're contained systems that they grow the sugarcane in, and they can vary the water table levels below them. And they have about 12 of these. And they're looking at the effects on sugarcane yield, soil properties, and nutrient leaching if they vary the water table level. So they have a 40 centimeter depth in May, and they can vary to 16 centimeters in different treatments in August. And they also flood and don't flood it. The actual treatments aren't important in what I want to tell you because we didn't see many differences. Just want you to understand what the project was. For this one, we actually had 8 DEA treatments. It was easier for us to do because there was only 12 lysimeters versus 48 seep sites, so we could run more samples. So we added nitrogen, glucose, and then we had a glucose and nitrogen. So you had your nitrogen, carbon, and a carbon-nitrogen source, and then we did that acetylene block where you get that total. So we actually had 8 treatments so that we could look at what were the effects of nitrogen and carbon. Now we were expecting, because these are highly organic soils with a whole lot of carbon in them, that you wouldn't see any effects from the carbon compared to the nitrogen. These were the greenhouse gas emissions. There's that photoacoustic gas analyzer measuring emissions out of there. We really didn't see much. Again, the variability in these treatments, even with moving the water table around, and here's your water table treatments, there's no difference in nitrous oxide emission. The variability is just too great. Carbon dioxide was a little higher in the 40-40, which was flooded occasionally, which might just indicate that moisture coming in and out helps keep the microbes active. When you come down to temperature differences from April to September, it's a little more obvious for some reason. N2O is a lot higher in April, maybe due to fresh mineralization of nitrate that could be denitrified. And then CO2 was highest when it was warmest in July, which makes sense for microbial respiration. So we didn't see much with the treatments. So flooding didn't affect that much, water table. The whole point of me showing you this data is where carbon becomes important in denitrification. We know nitrate's important, and we know moisture is important from what we saw in the seep data. But what we saw in Florida is even though these were highly organic soils, this is denitrification. This is with nitrate added, this is with glucose added, and this is glucose and nitrate. So the glucose has a larger effect, particularly when combined with nitrate, than nitrate by itself. Especially down here when you come to the total. You have A, B, C, D. So glucose and nitrate combined together produce the most DEA, so you maximize it, but glucose by itself had a larger effect than nitrate. And this kind of surprised us because we thought we were coming into highly organic soils, that there would be enough carbon that they would be nitrogen starved. But what we actually found out is that they were carbon starved. And this comes back to that thing I mentioned about carbon quality, fulvic acid versus humic acids. So if these soils were drained 50 years ago, the organic matter that's going to be mineralized first is that fresher carbon source. And you end up with a higher C to N type material, something that has lower quality. So although there was plenty of carbon, they obviously, from what we saw here, didn't have access to it. So in this case, adding carbon to these soils increased denitrification. So I have a little more data, which isn't showing up in these graphs, but it doesn't matter. Over here, I don't know where the graphs went, but this was actually incomplete denitrification, and you can see these letters climbing up, and complete. Incomplete was very low. The N2O that came off these soils was incredibly low. It didn't even match when you added nitrate and glucose and looked at total. So N2O emissions off these soils was pretty minor. Everything was going all the way to N2. And again, when you added glucose and nitrate, you saw the most additions. And the other two graphs I had here just showed that there weren't any differences in any of these treatments. So what we went looking for, does the variation in the water table level affect denitrification? If it's there, we didn't have enough data to prove it. So at this point, to answer any questions you guys have, denitrification is a really variable reaction. It's hard to predict in terms of values. To me, the research that I did pretty much showed me that trying to say how much was coming off the soil or how much denitrification occurring was difficult, but it was a lot easier to look at relative changes between soils. To say a wetter soil has more denitrification than a dry soil was easy, versus actually trying to predict how much was going to come off of a soil. So if there's any questions, I can answer them now.